I thank the organizer for the opportunity to talk about malaria immunity. And also, maybe as an introduction, I'm going to debunk the news that we have a malaria vaccine, even if we have a malaria vaccine which has been accepted by WHO, but this is a poor malaria vaccine. And if it was the same for COVID, we will be all dead by now. So I'm going to talk about malaria and immunity, and in this talk I will try to convince you that we need to do more work and identify antigens which will be the basis for a real and efficient malaria vaccine. So I'm not going to go too much into the details of the cycle that has been presented by the previous speakers. The malaria cycle is complex, going from the mosquito to the liver to the blood, and there are many part of the cycle that you could target to break the cycles. You could try to break the infection to the liver. You could try to eliminate the parasite from the liver. You could try to eliminate the parasite from entering into red blood cells. You could try to eliminate the infected red blood cells, and you could also try to block transmission to the mosquito. So it gives you many different ways of blocking, many different targets, but also I like the complexity, because since the parasite will be either intracellular or extracellular, you will have to design different ways to target these different forms of the parasite. So, as I said, there is report that there is a vaccine for malaria which has been accepted by WHO, but in fact we have a malaria vaccine for many years that it's called the irradiated sporozoite, and it targets the first stage of the infection, prevent the parasite, the sporozoite, to enter into the hepatocyte. And this is a very potent system, and it has been shown first to work in mice by Rtss Vaccine in the 60s in New York, and have been further demonstrated in rats, monkey, and humans. But at that time, in the 70s, when they did the experiments to induce protection, even if it was done in a small number of individuals, it was 4 or 5 per groups, you needed to submit these people to over 1,000 mosquito bites over a 6-month period. So even if we had a good vaccine, experimental vaccine, I would say, this was not feasible, because you cannot do mass production of mosquito and submit the volunteers to that numbers of bites over such a long period. However, this irradiated sporozoite model was a great tool to decipher protection against at least the sporozoite stage and the liver stage. So it was shown that this immunization system could induce strong immunity against the sporozoite and the liver stage, but none against the blood stage. And it helped to identify the first antigen which was cloned for malaria, which was called the circumsporozoite protein, which cover the whole surface of the sporozoite. And this identification of this antigen was the basis for the next 20 years of work to develop a vaccine based on the CSP. And that was the achievement in the 90s, where a group in Holland and then in the army, the Walter Reed, together with GSK, developed these vaccines called the Rtss Vaccine, which in fact is the construction where you have the circumsporozoite protein, part of it, the middle part and the C-terminal part, which is fused to the hepatitis B antigen. And the advantage is that they form some kind of polymers, which is very immunogenic. And the first results, which is now more than 20 years ago, 25 years ago in human, were very encouraging. In soldiers, volunteers, protection was around 70 to 80%, and that was very encouraging at that time. And when these trials were brought to the field, first in Africa in adult, the protection, the sterile protection, meaning that when you are infected, you don't develop a parasite in the blood. So this sterile protection was 48% in field condition. However, protection didn't last more than 6 months. And then there were many more field trials. I think we are around the 20-25 now, from different category of population, from adults to infants to children. And what we get for all these trials, which now I think encompass more than 200 million people, is that protection, you could obtain protection, but it's variable. And it's usually from 8 to 50%. And in children, it's in infants, it's 8%. So not at all efficacious for the population, which is the most at risk to die of malaria in Africa. And another important aspect is that immunity is short-lived. It lasts at best 6 months to 9 months, depending on the studies. And then you could do a recall immunization, but again, you will not extend for more than a year the protection. However, as I mentioned in my introduction, this was the first malaria vaccine license, and a few months ago it was supported by WHO. So as I say, for me, it's not a very good vaccine. A vaccine which lasts for 6 months and have a limited efficacy is not what I would expect. And one of the main cause of failure is that, in fact, the vaccine is strain specific. Most of the efficacy of this vaccine is linked to the production of antibody against the CSP. And the people who have high CSP level are usually, these high CSP levels are usually associated with protection. However, to make good antibodies, you need to have good T cells, and especially what we call the T helper T cells or CD4 T cells. And in this study done in 2016 by the group in Harvard by David Nevsky, they shown that people who have breakthrough infection have a parasite which contain a mutation into the CD4 epitopes. So this means that these parasites have escaped T cell recognition, and since the T cells could not be stimulated, they could not provide sufficient help to make the good antibody response. And so this raises another issue, which is that if you select by vaccination escape parasite, progressively this parasite, which could go normally and transmit to the population, will be the predominant population. And then your vaccine will have decreased efficacy over time. So another argument for me to say that the Rtss is not the best vaccine that we have. So nevertheless, this didn't prevent people to go back to, I would say, to the basic. We have irradiated sporozoite that we know works in humans. And there is a company called Sanaya in the US who said, okay, since the subunit vaccine have nearly all failed to give protection, why we don't revisit irradiated sporozoite in human? And so the idea was to mass product sterile sporozoite that they could freeze at use when you want, at will, and then they will use this product now to immunize people. So they first did a trial when they injected volunteers into the muscle, and the protection was very bad. And when they compare and they realize that in fact the immunity given by irradiated sporozoite when you use mosquito as the injecting factor, it was like the sporozoite will inject, the mosquito will inject the sporozoite into the body, and then the sporozoite which are inactivated by radiation but are still mobile could go to the liver, and even if they cannot grow more in the liver, they still go there. And so they say, okay, it's because the sporozoite are going to the liver. So they repeated the experiment now by, instead of injecting intramuscularly sporozoite, they injected the parasite intravenously, which was a bit of a challenge because no vaccine are injected normally through that route. And to what was, I would say, expected, now they could reach a protection, complete protection, and complete sterile protection in human using irradiated sporozoite. So they have been repeating this experiment in field condition in Tanzania, in Kenya, and in Mali. And contrary to what happened to healthy, clean volunteer, in Africa, the protection is always lower. It's between 30 to 60%. So there might be two reasons for that. The first one, they use one parasite strain to make their product. And in the field, there are multiple strains of parasites. So you have polymorphism, and some could escape the protection or the immunity given by the original product. And the other, that in Africa, you are frequently infected by other parasites, but other diseases, and in particular by worms. And it has been shown that worm infection tend to tolerize immune responses. So, but in any case, in adult, you still have protection, but it was a reduced one. And a recent article published, I think 2 months ago, showed that as for Rtss, this system, irradiated sporozoite, doesn't work into infant. They were not protected. So there are much more to learn in terms of what happened in the field condition into the immune system, and why it's very difficult to translate what you do in a clean lab in Europe or in the US to a real world. So why there is no efficient malaria vaccine? As I said, the parasite has multiple strains and has been with us for years and years, and he has developed many escape mechanisms. And I could list a few here. Many antigens are polymorphic. The parasite could induce immunosuppression, and even like for dengue, for example, it could use antibody to its own profit, like the mechanism called enhancement. The parasite, compared to bacteria or to viruses, are the very large genome with 14 chromosomes and more or less 5,000 genes. And as I mentioned, all the work has been done, in fact, in a handful of antigen. I would say less than 50. And so which antigen is the good one? Have we identified the good antigen? And are we putting all our money in the right one? And just to mention, we, long time ago now, did an experiment where we demonstrated, in fact, that if you use irradiated sporozoite, the mice could be protection independently of the circumsporozoite protein. So indicating that there are other antigens which are important in inducing immunity and it was in animal immunized with irradiated sporozoite. And as an immunologist, I'm always also asking is, what is really the mechanism that we should target? I indicated before that the parasite has different form, different localization. And so it will be, depending on the form and localization, subject to different immune response. If the parasite is intracellular, using antibody make no sense. If the parasite is in extracellular, so you will need both antibody and CD4 T cells. So for every part of the cycle, you need to define which is the protective immune mechanism. And if you get this protective immune mechanism right, then you could look for the antigen and design a good molecule. So for many, and I always say that I have been doing malaria vaccine research for 30 years. It's a long, frustrating journey, and you need to be very, very resilient, because you know that most of our studies are going to end up in failure. So, as I mentioned, irradiated sporozoite is a good approach, but in the lab, and is not, even its Sanaya claim it's feasible, it's not really now is it for mass deployment. And subunit vaccine have been given really disappointing results for many years. So a while ago, we went back to the board and say, okay, if live parasites are giving protection, can we design a new vaccine scheme that will be a bit different than irradiated sporozoite, but will induce the same level of protection? And for that, we use another approach where we use live parasite to immunize. We started in mice to immunize animals, and we stop the infection by treating with chloroquine, which is a common anti-malarial drug. And to do that, if we use sporozoite, we know that the parasite will infect the mice, go into the liver, and when he exits in the blood, he will be killed by the drug. And if we use a blood stage as the immunogen, you will have the beginning of infection, which will be blunted. And for both systems, in fact, if you do a drug treated animals after a challenge with the parasite, and then you re-challenge, you could induce complete sterile protection. And we showed that this immunity was not limited to one particular stage. We could block immunity at the level of the pre-erythrocytic stage, sporozoite, liver, or at the level of the blood stage. And even it was even, I would say, beautiful because the immunity was targeting all the stages. And so to make a long story short, we also identified which immune mechanism were acting. It was both antibody and T cells. And again, we asked the first questions, since most of our study were done with live sporozoite. What is the antigen involved? Is the circumsporozoite, which cover the sporozoite, involved? And to our surprise again, we could induce protection in absence of circumsporozoite protein. So really saying that other antigens were involved in protection. So that's okay. Doing that, all experiments were done in mice, and we say, okay, now can this be translated into human? And we were lucky to start a collaboration with Professor Sarah Vine from the Radboud University of Nijmegen, who have established a very nice system where they have a mosquito colony that they could infect with P. falciparum, and they could do human challenge with volunteers. And so we designed this protocol together where we submitted volunteers to 3 rounds of mosquito bites, 10 mosquito bites. And in between every round of mosquito bites, the people were under chloroquine. And after the 3 rounds of immunization, we waited for a month to let the chloroquine disappear, to not have confounding results. And then we challenged these volunteers with 5 mosquitoes, and 5 mosquitoes, which corresponds to less than 50 mosquito bites. And we had a control group which were submitted to the same regimen, except that the mosquito were not infected. And to our delight, I would say, we obtained complete sterile protection, meaning none of the volunteers which received the infected mosquito bite under chloroquine prophylaxis developed infection. And you could see here on the right, all the volunteers which received non-infected mosquito bites were infected. And so that was very encouraging. This has been repeated now for more than 300 people in Nijmegen, but also in the NIH in the US. And in fact, it was shown that the protection works very, very well when you immunize and challenge with the same parasite. But if you do challenge with an heterologous strain, you have a reduction of protection of 50%, but you still have 50% protection. And what also was really interesting is that if you re-challenge 29 months after people have been followed, we will still have more or less 4 out of 6 people protected, which really indicate that this protocol induces a long-term immunity. It has been now done in field conditions, and the protection was still very high, around 60 to 80% in experiments done in Africa by the group of Steve Hoffman. Okay, so my lab was interested of trying to now understand in human what was the mechanism. So, and especially what are the antigens which are recognized by this immunization with the optic, the idea of using that to make a vaccine that could be more amenable for mass deployment than using live sporozoite immunization. So first, we focus on the antibodies, and we tested if the antibodies from the protected versus the non-protected could inhibit the invasion of the sporozoite into a hepatocyte, human hepatocyte in vitro. And if you look at the lower bar here, in fact, we could reduce significantly the number of parasites developing into an hepatocyte if you use serum from people who have been immunized. And it was further shown by the group of Robert Sauerwein that in a humanized mouse model where human hepatocytes are grafted into the liver of mice, immunodeficient mice, that also the passive transfer of immunoglobulin protect against sporozoite challenge in this model. But the question is, which is the antigen recognized? So in the lab, we started a study now for the last 10 years where we created libraries of antigens that we suspected might be involved in protection because they are expressed on the surface of the sporozoite or at the beginning of the development in the liver. So to do that, we cloned this antigen into a vector, and which allows the expression of this antigen on surface of a cell. And so we tagged with little sequence so we could check for the quality of expression on the surface. And having these cells, now we could come with an antibody specific for the tag and see the level of expression. And this is shown here. If you look at in fact, the lower quadrant, the lower quadrant on the right here, you could see that using the two tags, we have a very high expression of our antigen of interest. And now, if we use antibodies coming from the people either who have been infected with malaria or who have been immunized, you could see again in the lower quadrant here, we're using a hyperimmune serum. This serum came from Tanzania in people who have been submitted from multiple rounds of infection, that you could detect expression of this antigen, and this allows us to quantify the level of recognition by antibodies in the serum. And so we could calculate what we call a serum response. And doing that, and I'm sorry because the graphs are quite small, but we could validate expression of our antigen, test a large number of antigen and serum, and to our interest, we found that 3 antigens, PF Mabel, PF Trap, and PF SEA1, were recognized specifically by people who have been immunized with the sporozoite under chloroquine. And so we went further and tested in vitro antibodies specific for the Mabel, and could see that antibody against different constructs could really inhibit the development, you see the 2 bars here, the development of sporozoite into the hepatocyte, really telling us that Mabel is one of the antigen of interest and maybe one of the main targets for immunization. And we verified that into the mouse system. And so my first part, so we have shown that using this system that there is, you could immunize and protect animals but humans, and this allows using the serum that you could get into the system to protect, to identify the antigen which may be involved in protection. So in my 5 minutes left, I will just go briefly on the new antigen that we have identified using the same system, because we keep adding. I told you in the introduction that there are more than 5,000 genes in the genome, so we keep adding new genes into our screen, and we identify here this antigen that we call PFFT1, and we compare on the right here people who have been immunized with people protected and people who are not protected, and you could see that there is an increase of recognition, and this antigen is associated with protection. We determined that this antigen, contrary to this other antigen that we showed, is recognizing the blood stage, and that the staining here in red. And when you look at the expression during the blood stage, we see that it's also corresponds at the time when the merozoites are formed into the red blood cells here. And to really go further and characterize this antigen, we created parasites which are knockout for this antigen. You could see here there is no more expression of the mRNA of this antigen here, and we look at the characteristic of the knockout parasite, and we saw that they grow nearly follow the same pattern of growth, but they were less. They grow less well in vitro compared to the wild type parasite, and in fact they had always a delayed in growth compared to the wild type parasite. And now we also develop specific antibodies against this new molecule and tested if they were able, because it's a blood stage antigen which is not expressed in the liver, where this antibody could have an effect. And so we tested the capacity to agglutinate the sporozoite here, and you could see that when you have the antibody, you increase agglutination. We tested antibody ability to block invasion in the red blood cells, and you could see also that we had quite a good inhibition. And we tested also the ability of the antibody to block what is called egress, which is the release of the merozoite from the red blood cells. And again, this was shown here, and we had a strong inhibition. And we also look for phagocytosis, which is another mechanism of action of antibodies. And we showed that infected red blood cells were phagocytosed. Merozoite isolated were also phagocytosed very actively by monocytes, macrophages. And another mechanism called antibody-dependent cell inhibition, where antibodies stimulate monocyte to kill the parasite, was also induced by the type of antibody recognizing this antigen. And the last mechanism that we look at is the capacity of complement together with antibody to lyse merozoite. And we show that different parts of the complement system were able to bind together with antibody to the merozoites, recruit the lytic factor of the complement, and kill the parasite here, the merozoite. So I hope that I have convinced you that we have identified a new antigen which is linked and with protection. And what is very interesting for this antigen is that it's only expressed for falciparum and the monkey equivalent, called, which in fact chimpanzee, which is called Reichnoi. You cannot find it in other parasite species. And we found that many mechanisms where these antibodies recognizing this parasite could inhibit the blood stage. So we have much more work to do to really look at the importance of this antigen in protection into natural infection. And this work included a lot of people, past and active member in the lab, and my collaborator from the Radboud University, where we did the human challenge and group in France and in NTU. And I thank you for your attention.
Thank you very much, Laurent. Very nice talk.